Good day everyone. Today we'll be discussing DNA replication, transcription, and translation. So first, let's proceed with DNA replication. So for transmission of genetic material from parent to offspring and from cell to cell to occur, the genetic material must be copied. So in this process, the original DNA strands are used as templates for the synthesis of new DNA strands. So DNA replication relies on the complementarity of DNA strands according to the ATGC rule. So during replication process, two complementary strands of DNA come apart and serve as template strands or parental strands. So after the double helix has separated, individual nucleotides have access to the template strands. So the hydrogen bonding between the individual nucleotides must obey the ATGC rule. So to complete the process, a covalent bond is formed between the phosphate of one nucleotide. So after the process, two newly made strands are referred to as the daughter strands. So there are three possible models for DNA replication. So we have the conservative model, the semi-conservative model, and the dispersive model. So we'll tackle each one of them. So for the conservative model, both strands of the parental DNA remain together following the DNA replication. So the original arrangement of the parental strands is completely conserved, while the two newly made daughter cells also remain together with the following replication. For the semi-conservative, the double-stranded DNA is half conserved. So the newly made double-stranded DNA contains one parental strand and one daughter strand. So it is also known as the correct model. For the dispersive model, so the segments of the parental DNA and newly made DNA are interspersed in both strands following the replication process. So an experimental approach to reveal how DNA replicates was first suggested in 1941 by J.B.S. Haldane. In 1958, Matthew Meselson and Franklin Stahl tried Haldane's experiment using E. coli bacteria. So their density shift experiments involved labeling DNA with a heavy isotope of nitrogen. So steps of DNA replication. So when DNA replicates, it unwinds, breaks, and builds a new nucleotide chain and mends. So there's an enzyme in the steps of DNA replication that is helicases. So they hold apart replicating DNA so that other enzymes can guide the assembly of a new DNA strands. So helicases can also repair errors in the replicated DNA. So human DNA replicates about 50 bases per second, and the site where DNA is locally opened, resembling a fork, is called a replication fork. So saan nagsisimula yung steps of DNA replication? It begins with the helicase breaks the hydrogen bonds, then the binding proteins hold the two strands apart, then another enzyme, yung primase, ito naman yung nag-a-attract complementary RNA nucleotides to build a short piece of RNA. Yung short piece ng RNA na yun, they are called RNA primer. So that RNA primer is required. Bakit? It is the major replication enzyme. The DNA polymerase can only add bases to an existing nucleic acid strand. So polymerase naman is an enzyme that builds a polymer, which is a chain of chemical building blocks. So after that, the RNA primer attracts the DNA polymerase, which brings in DNA nucleotides complementary to the exposed bases onto the parental strand. So this strand serves as the model or the mold or template. So new bases are added one at a time starting at the RNA primer. So the DNA strand grows as hydrogen bonds from between the complementary base. DNA polymerase works directionally, adding new nucleotides to expose 3' end of the sugar in the growing strand. So overall, replication proceeds in a 5' to 3' direction because this is the only chemical configuration in which the DNA polymerase can add bases. So ligase come from the Latin word meaning to tie, is an enzyme that then seals the sugar phosphate backbones. So these pieces, up to 150 nucleotides long, are called the Okazaki fragments. So this is the summary of the enzymes involved in the DNA replication. So ito na yung tandaan yung table because it's very important. Baka matanong sa mga quizzes natin. Helicase unwinds parental double helix. The binding proteins stabilize separate strands. The primase add short RNA primer to template strands. The DNA polymerase binds nucleotides to form a new strands, while the ligase, they join the Okazaki fragments and seals other nicks in the sugar phosphate backbone. So this is the summary of the DNA replication. First, yung parent DNA molecule, di ba meron two complementary strands of base paired nucleotides. Tapos yung second doon, nag-unwind sila at nagse-separate. Dito, nag-unwind sila, nagse-separate. Tapos, yung each parental strands provides a template. Yung naghiwalay, yun yung magiging template. Tapos, after nun, the sugar phosphate bone of the daughter strands close, forming new daughter strands ng DNA. So, let's proceed sa DNA transcription and translation. So, the central dogma. So, the central dogma of genetics, it is also known as the central dogma of the molecular biology. It was first enunciated by Francis Crick. So the flow of genetic information from the DNA to mRNA and to the polypeptide. So ito yon, starting tayo sa DNA replication. So the DNA mix copy then are transmitted from cell to cell and from parent to offspring. So the chromosomal DNA, it stores information in units called the genes. So in transcription, it produces the RNA copy of a gene. So ang magiging RNA copy na yun yung mRNA or the messenger RNA. It is the temporary copy of a gene that contains information to make a polypeptide. Tapos, translation, it produces a polypeptide using the information in the mRNA. So the polypeptide becomes part of the functional protein that contributes to an organism's trait. Yan yun, we have the replication, tapos transcription, tapos translation. So the DNA replication preserves genetic information by giving each new A cell a complete set of operating instructions. So protein diversity in the human body. So these are different proteins. Please, lahat ng mga tables, please makikitanda po. First, we have the actin, myosin, and dystrophin for muscle contraction, antibodies, antigens, and cytokines for immunity, carbohydrates, lipases, proteases, and nucleases for digestion. They are the digestive enzymes. Casein is a milk protein, and collagen, elastin, and fibrillin for connective tissue. Next, we have the colony-stimulating factors and erythropoietin for blood, DNA and RNA polymerase for DNA replication, gene expression. Ferritin, we have iron. Fibrin and thrombin for blood clotting. Growth factors, kinases, and cyclins for cell division. Hemoglobin and myoglobin for oxygen transport. Insulin and glucagon for control of blood glucose level. Keratin for hair structure. Tubulin and actin for cell movements. And tumor suppressors for cancer prevention. So, to use the genetic information in the nucleus, the process of transcription first copies a gene into a RNA molecule that is complementary to a strand of DNA double helix. While the process of translation uses information in the three types of RNA to manufacture a protein by aligning and joining specified amino acids. So transcription is the ability of the genes to produce an organism's trait, relies on the molecular process of gene expression. So during transcription, the gene sequence within the DNA is used as the template to make a complementary copy of RNA. So the RNA is the bridge between the gene and the protein. It is synthesized against to one strand of the double helix called the template strand with the assistance of an enzyme, the RNA polymerase. The other strand of double helix is the coding strand. So RNA is the ribonucleic acid. It is a single strand. Anong sugar niya? Ribose. Ang mga bases niya, we have the adenine, uracil, guanine, and cytosine. Compared to DNA, na thymine, yung base pair, nakapartner ng adenine, sa RNA, napapalitan siya ng uracil. So there are three major types of RNA. First, we have the messenger RNA, ribosomal RNA, and the transfer RNA. So first, let's proceed to messenger RNA. So it carries the information that specifies a particular protein. So each three mRNA bases in a row form a genetic code word or the codon. So most mRNAs are 500 to 4,500 bases long. Ulit, yung 3 mRNA bases in a row, ang tawag doon is codon. Pag sinabi namang ribosomal RNA, they range from 100 to nearly 3,000 nucleotides long. So ribosomal, ibig sabihin ribosome. So the ribosome, di ba, structural support for protein synthesis. Pag transfer RNA naman, meron silang 75 to 80 nucleotides long. It binds an mRNA codon at one end and a specific amino acid at the other. So, it always carries the same amino acid. So, there are three stages in transcription. We have the initiation, elongation, and the termination. First, the initiation or the promoter. It is a special sequence that signals the start of a gene, functions as the recognition site for transcription factors. So yung transcription factors enables RNA polymerase to bind to the promoter. So in genetics, the first transcription factor to bind called the TA binding protein or the TATA binding protein. It is attracted to the DNA sequence called the TATA box or the TATA box. The base sequence, TATA surrounded by long stretch of guanine and cytosine. The coming together of these components is called the transcription initiation. Elongation naman, the RNA polymerase slides along in the DNA in an open complex to synthesize protein. For the termination, meron naman siyang terminator. Ito yung nagdi-dissociate from the DNA. So ulit, the three steps of transcription, we have the initiation, meron dyan promoter, tapos elongation, at termination. Tapos the typical rate of the transcription in humans is 20 bases per second. So RNA is transcribed using only the gene's template strand. So to determine the sequence of the RNA bases transcribed from a gene, write the RNA bases that are complementary to the template strand. So for example, a DNA template strand that has a sequence of CCTAGCTAC is transcribed into RNA with the sequence GGAUCGAUG. And the coding DNA sequence is GGATCGATG. So magkaiba lang sila sa pag-prepare when it comes to the adenine. Kasi nga pag RNA, uracil ang kapartner ng adenine, while for the DNA sequencing ay thymine ang kapartner ng adenine. So previously nga, transcription is the process of making a RNA copy of the gene sequence. So this copy is called the messenger mRNA molecule. It leaves the nucleus and enters the cytoplasm where it directs the synthesis of protein which is encoded. Tapos after nung process na yun, let's proceed to the Translation. So translation is the process of translating the sequence of mRNA molecule to a sequence of amino acids during protein synthesis. So the genetic code describes the relationship between the sequence of base pairs in a gene and the corresponding amino acid sequence that it encodes. So in the cell cytoplasm, the ribosome reads the sequence of the mRNA in groups of 3 bases. Ano nga ulit yung 3 bases na yun? Yun yung codon. So the steps in translation, the ribosome binds to mRNA. The ribosome starts matching tRNA anticodon sequences to the mRNA codon sequence. So each time a new tRNA comes into the ribosome, the amino acid that it was carrying gets added to the elongating polypeptide chain. Then the ribosome continues until it hits a stop sequence. Then the polypeptide form into its native shape and starts acting as functional protein. So protein folding. So protein must fold into one or more specific three-dimensional shapes. So the folding occurs because of the attraction and repulsion of atoms. So protein folds saan daw? Sa endoplasmic reticulum. So there are 4 levels of protein structure. Primary, yun ay yung amino acid sequence in a polypeptide chain. Secondary, yan ay yung loops, coils, and sheets or other shapes formed by hydrogen bond. Tertiary, ito na yung madali lang tandaan, 3-dimensional forms. Then the quaternary, yun na yung mga protein complexes. Yan lang yun, picture. Another picture. Then various proteins assist in its precise folding. We have the chaperone proteins. So it stabilizes partially folded regions in their correct form. We also have the folding catalyst, help new chemical bonds to form as a final shape arises. Tapos, in case of a protein misfold, folding sensor activates a cellular reaction to stress called the unfolded protein response. So, ano tong proteasome? It is a tunnel-like multi-protein structure. So, through it, protein is stretched out, chopped up, and its peptide pieces degraded into amino acids. So, misfolded proteins, not destroyed, can cause diseases. First, we have the cystic fibrosis. So, it prevents encoded protein from assuming its final form. We also have Huntington disease. Extra glutamines in the protein huntingtin cause it to obstruct proteasomes, which eventually kills the cells. So misfolded proteins that clog proteasomes can also occur in Alzheimer's disease, amyotrophic lateral sclerosis, Parkinson's disease, and the Lewy body dementia. So another type of protein folding disorder arises in glycoproteins called prions. So disease called the transmissible spongiform encephalopathies result when one conformation of prion glycoprotein is infectious, which means that it causes others to misfold. So for our last table, we have the disorders, protein folding and aggregation, Alzheimer's disease. Ano yung misfolded protein? We have the amyloid beta precursor protein. We have the familial amyotrophic lateral sclerosis, misfolded proteins, the superoxide dismutase, Huntington disease, Huntingtin, Parkinson disease, alpha synuclein, the Lewy body dementia, and the prion disorders. We have the prion proteins.